Did you ever wonder how crime scene investigators manage to find those invisible fingerprints and, well, make them visible? It's not magic. It's actually some brilliant chemistry at work.
Yeah, one really powerful method uses a chemical called 9-hydrin. It reacts with these tiny compounds that are naturally in our sweat, amino acids.
Amino acids. And this reaction creates these striking colored compounds that investigators can then actually see.
Exactly, a classic bit of forensic chemistry.
So today, we're taking a deep dive into this fascinating world of amino acids, peptides, and proteins. Our mission really is to give you a shortcut to understanding their structures, their properties, how they're made, and their absolutely critical biological functions.
Think of it as your backstage pass to the molecular machinery of life. It's pretty amazing stuff.
Okay, let's unpack this, starting right at the beginning with the fundamental building blocks themselves.
Right, amino acids.
So amino acids, what makes them so unique?
Well, they're organic compounds, obviously, but the key thing is they contain both an amino group, that's the NH-NH2, and a carboxylic acid group, the dacious COH, both on the same molecule.
Okay, and in proteins, the ones that make up so much of us, these two groups are almost always separated by just one carbon atom, right?
Precisely. We call that the alpha, or acar carbon.
And that alpha carbon is usually chiral, like a tiny molecular hand.
Yeah, exactly. It has 4 different groups attached, making it asymmetric. Think left hand versus right hand. They're mirror images, but not superimposable.
Ah, okay, except for glycine.
Right, glycine's the exception. It's R group, the side chain is just another hydrogen atom, so no chiral center there, it's achiral.
It's pretty incredible, though, when you think that hundreds of amino acids exist out there in nature.
Mm-hmm.
But only 20 alpha amino acids are the main players found in proteins.
And those 20 only differ by that unique R group, the side chain. That's what gives each one its specific character.
And you mentioned something about L-amino acids.
Yeah, another interesting point. Almost all naturally occurring amino acids in proteins are L-amino acids. It's a specific spatial arrangement, sort of like how most natural sugars are D sugars. Nature has its preferences.
Okay, so structure-wise, they're defined by that alpha carbon and the R group. What are their properties? You hear they're amphoteric.
That's right. Amphoteric just means they can act as both an acid and a base. They can donate a proton, thanks to the carboxylic acid group, or accept a proton, thanks to the amino group.
And what does that actually mean for how they behave?
Well, it's fundamental. It leads directly to this key concept called the deuterion.
Deuterion. Sounds complicated.
It sounds fancy, but it's straightforward. At physiological pH, so around 7.4, like in your blood, the amino group grabs a proton, becoming NH3 plus seg positively charged.
Okay.
And the carboxylic acid group loses its proton, becoming COO, negatively charged.
Ah, so you have both a positive and a negative charge on the same molecule, but overall it's neutral.
Exactly. A net neutral compound, but with internal charge separation. It behaves like an internal salt.
And this deuterion thing, it affects their properties.
Massively. Because they exist in this charged deuterionic form, amino acids tend to have, well, surprisingly high melting points for organic molecules, and they're generally very soluble in water. Think about it. Salts usually dissolve well in water.
That makes sense. An internal salt explaining solubility and melting point. Okay. And this acid-base behavior ties into pK values too.
Absolutely. Most amino acids have 2 main pK values. You've got pK1 for the carboxylic acid group, usually around 2 to 3, so quite acidic.
Right.
And pK2 for the ammonium group, the NH3 plus RC, typically around 9 to 10, so much less acidic, more basic.
And so some have a third one.
Yeah, if the side chain itself is acidic or basic, like glutamic acid or lysine, it'll have its own pK value, a pKr.
Okay, and this leads to the isoelectric point, the PI.
Precisely. The PI is that specific pH value where the amino acid exists predominantly as the deuterion, where the net charge is 0.
How do you figure that out?
Well, for simple amino acids without an ionizable side chain, like alanine, the PI is just the average of pK1 and pK2. So for alanine, it's about 6.02.
Okay.
But if you have an ionizable side chain, like lysine with its extra amino group or glutamic acid with its extra carboxyl group, the calculation is a bit different. You average the pKa values of the similar groups.
Ah, I see. So for lysine, you'd average the 2 amino group pKas.
Exactly. And for glutamic acid, you'd average the 2 carboxylic acid pKs. It tells you the pH where that specific amino acid won't move in an electric field.
And that's crucial for electrophoresis?
You got it. Electrophoresis is a technique used to separate mixtures, often proteins or amino acids, based on charge.
How does it work?
You place your mixture, say amino acids, in a buffered solution at a specific pH and apply an electric field using electrodes.
Okay.
Now, depending on an amino acid's pI relative to the buffer's pH, it will have a net charge. If the pH is below its pI, it'll be protonated, have a positive charge, and move towards the negative electrode, the cathode.
And if the pH is above its pI?
Then it'll be deprotonated, have a negative charge, and move towards the positive electrode, the anode. The further the pH is for the pI, the greater the net charge, and the faster it moves.
So they separate out based on their pI values and the buffer pH.
Precisely. It's a really powerful separation method.
And connecting back to the start, this is how 9-hydrin helps with fingerprints after separation.
Exactly. After you run the electrophoresis, you can spray it with 9-hydrin. It reacts with those separated amino acids wherever they ended up, and boom, you get those visible purple spots.
Wow. So the pattern of spots tells you something about the amino acids present.
It can, yes. The number of spots indicates the number of different amino acids, at least those that were present in sufficient quantity in the fingerprint residue.
Okay, so their charge properties are key for detection and separation, and also for life itself. Which brings us to nutrition. We need these things, right?
Absolutely. Of those 20 common amino acids, 10 are considered essential for humans, meaning our bodies can't make them from scratch or can't make them fast enough. We absolutely must get them from our diet.
Right, and that's where complete and incomplete proteins come in.
Exactly. Complete proteins, like those found in meat, fish, eggs, dairy, they contain all 10 essential amino acids in roughly the proportions we need.
Okay, and incomplete.
Incomplete proteins, typically from plant sources like rice, corn, beans, are low in or lack one or more of the essential amino acids.
So for vegetarians or vegans?
They need to be mindful of combining different plant protein sources like beans and rice, for instance. This complementary diet approach helps ensure they get the full set of essential amino acids over the course of the day. It's all about getting those necessary building blocks.
Okay, we know what they are, why they matter. Now, how do chemists actually make them in the lab?
Good question. Historically, several methods were developed. You have things like the Helvall-Hard-Zielinski reaction applied to acids, the amidomalonate synthesis, and the Strecker synthesis.
What do those involve, briefly?
Well, the Helvall-Hard-Zielinski approach involves first putting a bromine atom on the alpha carbon of a carboxylic acid, then replacing that bromine with an amino group using ammonia.
Okay.
The amidomalonate synthesis is kind of a clever twist on a classic reaction called the malonic ester synthesis. You start with a specific reagent, diethylacetamidomalonate, deprotonate it, add an alkyl group that defines your side chain, and then hydrolyze and decarboxylate to get the amino acid.
And the Strecker synthesis?
That starts with an aldehyde. You react it to form an intermediate called an alpha-aminonitrile, and then you hydrolyze the nitrile group to get the carboxylic acid. The aldehyde you start with determines the amino acid you end up with.
But these methods, they have a drawback.
Yes, a significant one for many applications. They typically produce a racemic mixture.
Meaning an equal mix of both the L and the D forms, the mirror images.
Exactly, a 50-50 mix.
But you said earlier that nature mostly uses the L form.
Right. So if you need just the L amino acid, say for making a drug, getting a racemic mixture isn't ideal. Separating enantiomers, what we call resolution, can be done, but it's often inefficient. You're essentially throwing away half your product.
That sounds wasteful.
Yeah.
So how do chemists get around that? How do they make just one specific enantiomer?
That's where the field of enantioselective synthesis, or asymmetric synthesis, comes in. It's a much smarter approach.
How does that work?
It involves using chiral catalysts. These are catalysts that are themselves chiral, handed, and they guide the reaction to preferentially form one enantiomer over the other.
Ah, like a template.
Sort of, yeah. Think of William S. Knowles, who won a Nobel Prize for this work. He developed methods for asymmetric hydrogenation using chiral catalysts, famously used to synthesize L-DOPA, a key drug for Parkinson's disease.
And you could get mostly the L form.
Yes, very high enantiomeric excess, often abbreviated E. Like 99% E means you get 99.5% of the desired enantiomer and only 0.5% of the other. It's incredibly powerful for making pure single enantiomer compounds.
So chiral catalysts are the key for L amino acids, except glycine, I guess.
Right, good point. Glycine isn't chiral, so you don't need an antioselective method for it. You just make glycine.
Okay, so now we have our amino acid building blocks, maybe even the specific L form we need. How does nature link them together to build peptides?
Nature links them using amide linkages, which in this context we specifically call peptide bonds.
How is that bond formed?
It's essentially a condensation reaction. The carboxylic acid group of one amino acid reacts with the amino group of another, and a molecule of water is eliminated, forming that CN-amide bond, the peptide bond.
And there's a specific way we write these out.
Yes, a very strict convention. Peptides are always written or drawn with the N terminus on the left.
That's the end with the free amino group, the NHNH3+.
Correct. And the C terminus on the right.
The end with the free carboxylate group, the NACH-EOO.
Exactly. And the order is absolutely crucial. Allyl, alanine followed by glycine, is a totally different molecule, a different dipeptide, than glyala, glycine, followed by alanine. Sequence matters.
Okay, so the peptide bond links them. Does the bond itself have any special characteristics, geometry-wise?
Oh, definitely. This is really important for protein structure later on. Because it's an amide linkage, there's resonance.
Meaning electrons are shared across the OCN system.
Precisely. This gives the CN bond significant double bond character. It's not a full double bond, but it's stronger and shorter than a typical CN single bond.
And what does that do?
It restricts rotation around that specific peptide bond. The 6 atoms involve the alpha carbon, the CO group, the NH group, and the next alpha carbon all tend to lie in the same plane. It creates a planar unit.
So the whole chain isn't floppy around that bond.
Not around the peptide bond itself, no. There's still free rotation around the bonds to the alpha carbons, which allows the overall chain to fold, but the peptide bond unit itself is rigid and planar. This planarity is a major constraint on how proteins can fold.
Interesting. So planarity from resonance. What about other special features? Disulfide bridges.
Ah, yes, disulfide bridges, very important. Remember cysteine. It's unique among the 20 common amino acids because it has a thiol group, dis-SH, in its side chain.
Right.
Well, 2 cysteine residues, which might be far apart in the linear sequence but come close together when the protein folds, can react under oxidizing conditions. Their thiol groups lose hydrogen, and the 2 sulfur atoms form a covalent bond, an NSS bond. That's a disulfide bridge.
Like a staple holding parts of the chain together.
Exactly. It's a covalent crosslink. These bridges can form within a single polypeptide chain, which we call intrastrand, or between 2 separate chains, interstrand.
And they affect the 3D shape.
Greatly. They add significant stability to the folded structure of many peptides and proteins, locking them into their functional conformation. Think about hair perms that involves breaking and reforming disulfide bridges in keratin.
Right. Okay, so peptides are chains of amino acids linked by peptide bonds, sometimes with disulfide bridges. What do they actually do biologically?
They have incredibly diverse roles. Some act as hormones, others as neurotransmitters, some have antibiotic properties. They're not just intermediates on the way to proteins. Many peptides have vital functions themselves.
Can you give some examples?
Sure. Take the enkephalins, leucine enkephalin, and methionine enkephalin. They're small pentapeptides, just 5 amino acids long, found in the brain.
What do they do?
They act as natural painkillers, binding to opioid receptors. Interestingly, they only differ by the very last amino acid at the C terminus, yet both are involved in pain control.
Wow. Any others?
Well, there's bradykinin, a nonapeptide, 9 residues that acts as a hormone to dilate blood vessels and reduce inflammation.
Okay.
Or think about vasopressin and oxytocin. These are also nonapeptides, structurally very similar, differing by only 2 amino acids, but with vastly different functions.
How different?
Vasopressin, also called antidiuretic hormone, regulates blood pressure and water reabsorption by the kidneys. Oxytocin, on the other hand, is famous for inducing labor during childbirth and stimulating milk production.
That's incredible. Such a tiny structural difference, such a huge functional difference.
It really highlights a key principle in biochemistry. Structure dictates function, sometimes down to the smallest detail.
And you mentioned antibiotics, too.
Yes, some peptides are potent antibiotics. You might recognize names like bacitracin A or polymyxin B from topical ointments like Neosporin.
Oh, yeah.
These are often cyclic peptides, sometimes containing unusual D-amino acids not typically found in humans. They often work by disrupting bacterial cell wall synthesis, essentially making the bacteria burst.
Okay, so peptides are important functional molecules. But how do scientists figure out their sequence? If alagly is different from glyala, knowing the exact order must be critical.
Absolutely critical. Determining the primary structure of the amino acid sequence is fundamental. One classic method is called Edmond degradation.
How does that work?
It's a clever, stepwise process. You chemically label the N-terminal amino acid, then specifically cleave it off the rest of the peptide chain without breaking other peptide bonds. You then identify that cleaved-off, labeled amino acid derivative, usually something called a PTH derivative. Then you repeat the cycle. Label the new N-terminal amino acid, cleave it, identify it, and so on.
So you identify them one by one from the N-terminus.
Exactly. It's often automated now and works well for peptides up to about 50 residues long.
What about longer peptides, or even proteins? 50 residues isn't very long compared to a full protein.
Right. For larger peptides or proteins, Edmond degradation alone becomes less practical because tiny amounts of side products accumulate with each cycle, making the results messy.
So what's the approach then?
You use enzymes called peptidases or proteases. These enzymes act like molecular scissors, but they're very specific. They only cut peptide bonds next to certain amino acid residues.
Like specific scissors for specific amino acids.
Precisely. For example, the enzyme trypsin specifically cleaves the peptide bond on the carboxyl side, the C-terminal side, of basic amino acids like arginine, arg, and lysine. Another common one is chymotrypsin. It cuts on the carboxyl side of bulky aromatic amino acids like phenylalanine, phea, tyrosine, ter, and tryptophan, tryptophan.
So you use these enzymes to chop the big peptide into smaller pieces.
Exactly. You take your large peptide, treat separate samples with different enzymes, like trypsin in one tube, chymotrypsin in another. This gives you different sets of smaller fragments.
Then what?
Then you sequence each of those smaller fragments using Edmond degradation. And the key is, by comparing the sequences of the fragments generated by different enzymes, you look for overlaps.
Ah, like putting together a jigsaw puzzle.
Well,
The overlapping pieces tell you how the original fragments fit together.
that's a perfect analogy. By finding those overlaps, you can deduce the complete sequence of the original large peptide or protein. It's painstaking work, but very powerful.
Okay, that's sequencing. What about making peptides in the lab? Synthesizing them. You mentioned protecting groups earlier for single amino acids. Is it more complex for peptides?
It definitely is. The main challenge is controlling which amino group reacts with which carboxyl group. It's called the regioselectivity problem.
Meaning, if you just mixed two different amino acids, say alanine and glycine,
Yeah.
and added a coupling agent.
You wouldn't just get alagly.
Yeah.
You could also get gliala plus ala, and glycally, a mixture of potentially 4 different dipeptides. Not very useful if you want just one specific sequence.
Right. So protecting groups are the answer here, too.
Absolutely essential. You need to temporarily mask the amino group of the amino acid that will provide the carboxyl group for the peptide bond, and mask the carboxyl group of the amino acid that will provide the amino group.
So you protect the bits you don't want to react yet.
Exactly. We talked about BOC, terpbutoxycarbonyl, for protecting amino groups. It's put on using diterpbutyl dicarbonate.
And it comes off easily with acid, making gas.
Right. Trifluoroacetic acid, TFA, removes it, generating isobutylene and CO2 gas, which just bubble away. Very clean and efficient. Thiamoc is another common amino protecting group removed by base.
And protecting the carboxyl group.
That's usually done by converting it into an ester, like a methyl ester or a benzyl ester. Esters don't readily react with amino groups under the conditions used for peptide bond formation.
So the overall strategy for making, say, alagly would be
Protect the amino group of alanine, for example, with BOC. Protect the carboxyl group of glycine, for example, as a methyl ester. Then couple bacala with gliome using a coupling agent like DCC, dicyclohexylcarbodiamide.
What does DCC do?
DCC activates the carboxyl group of balala, making it reactive towards the free amino group of gliome, facilitating peptide bond formation.
Okay, so now you have bacala gliome, then you remove the protectors.
Correct. You'd remove the BOC group with acid, TFA, and then remove the methyl ester, often by hydrolysis with base, to get the final dipeptide, alagly. It's a multi-step, careful process.
That sounds like a lot of work, especially for longer peptides. Lots of purification steps in between.
It can be, especially with traditional solution phase synthesis. But this is where R. Bruce Merrifield's Nobel Prize-winning invention really revolutionized things. Solid phase peptide synthesis, SPPS.
Solid phase, what does that mean?
It's ingenious. You start by attaching the C-terminal amino acid, with its amino group protected, FD shape at bond, to an insoluble solid support, usually small polymer beads or resin.
So the growing peptide is physically stuck to these beads.
Exactly. Then you carry out the synthesis iteratively while the peptide is attached to the solid support.
How does that help?
It makes purification incredibly simple. After each step, say removing the FOMOC group or coupling the next FOMOC-protected amino acid using DCC, you just wash the beads.
Ah, so the excess reagents and byproducts just get washed away, but your peptide stays anchored to the beads.
Precisely. You remove the FOMOC group, wash. Add the next activated FOMOC amino acid, let it couple, wash, repeat, 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 adding one amino acid at a time.
That sounds much faster and cleaner.
It is. You only need to cleave the completed peptide off the solid support right at the very end. Merrifield famously synthesized ribonuclease, a protein with 124 residues. This way back in 1969. It was a landmark achievement.
124 amino acids. Wow. And this is automated now.
Oh yes, modern peptide synthesizers automate the entire SPPS process, allowing for the relatively rapid synthesis of quite long peptides and even small proteins.
Okay, so we can sequence them and synthesize them. Now, let's talk about proteins themselves. These are the really big players, right? How do we describe their structure?
We usually talk about protein structure on 4 hierarchical levels: primary, secondary, tertiary, and quaternary.
Okay, start with primary.
Primary structure is simply the linear sequence of amino acids linked by peptide bonds, including any disulfide bridges. It's the fundamental blueprint. Think of human insulin. Its primary structure consists of 2 specific chains linked by disulfide bonds. Knowing that sequence is the first step.
Got it. Sequence first, then secondary.
Secondary structure refers to regular, repeating patterns of folding within local segments of the polypeptide chain. Remember that planar peptide bond? That limits the possibilities.
Right.
The 2 most common secondary structures are the alpha helix and the beta-pleated sheet.
The alpha helix,
Right.
like a spiral staircase.
Exactly. It's a right-handed coil stabilized by hydrogen bonds between the CO group of one amino acid and the NH group of an amino acid 4 residues further down the chain. The R groups point outwards from the helix axis.
Any amino acids that don't fit.
Proline is often called a helix breaker because its cyclic structure and lack of an NH proton disrupt the regular H bonding pattern.
And where do we find alpha helices?
They're very common. A good example is a keratin, the main protein in hair, skin, and nails. Hair is almost entirely made of I helices bundled together.
Okay. And the beta-pleated sheet.
This structure involves segments of the polypeptide chain lining up side by side. They can run in the same direction, parallel, or opposite directions, anti-parallel.
So how are they held together?
Again, by hydrogen bonds, but this time the H bonds form between adjacent strands, linking the CO of one strand to the NH of the neighboring strand.
And the R groups.
They point alternately above and below the plane of the sheet, giving it a pleated appearance. Beta sheets are often found in proteins needing strength and rigidity, like fibroin, the protein in silk. Small R groups like glycine and alanine favor this structure.
So primary is sequence, secondary is local folding, like helices and sheets. What's tertiary?
Tertiary structure is the overall, unique, 3-dimensional shape of a single, entire polypeptide chain. It describes how those secondary structure elements, helices and sheets, and other parts of the chain fold and pack together in space.
What drives that folding?
It's driven by interactions between the R groups and the surrounding environment, usually water. The protein folds to achieve the most stable conformation. Typically, hydrophobic, nonpolar R groups get buried in the protein's interior, away from water, while hydrophilic, polar and charged R groups are exposed on the surface, interacting with water.
So it's like the protein tucks its oily bits inside?
That's a great way to put it. Disulfide bridges also play a crucial role in stabilizing the tertiary structure for many proteins. Myoglobin, the oxygen storage protein in muscle, is a classic example of a globular protein with a well-defined tertiary structure.
But this structure is delicate, right? Denaturation.
Very much so. Denaturation is the process where a protein loses its specific secondary and tertiary structure, its folded shape, without breaking the primary sequence, the peptide bonds.
What causes it?
Things like heat, extreme pH changes, certain organic solvents, or even vigorous shaking can disrupt the weak interactions, hydrogen bonds, hydrophobic interactions, that maintain the folded structure.
Like cooking an egg white.
Perfect example. The clear albumin protein denatures upon heating, unfolding, and aggregating into that familiar white solid. Importantly, denaturation almost always leads to a loss of the protein's biological function, because function depends critically on the correct 3D shape, and it's usually irreversible.
Okay, so primary, secondary, tertiary. There's one more. Quaternary.
Yes, quaternary structure applies only to proteins that are composed of more than one polypeptide chain. Each individual folded chain is called a subunit.
So it's how multiple folded chains fit together.
Exactly. Quaternary structure describes the arrangement of these subunits and the interactions holding them together to form the final functional protein complex.
Any examples?
The classic example is hemoglobin, the protein that carries oxygen in red blood cells. It consists of 4 separate polypeptide subunits, 2 alpha chains, and 2 beta chains, all packed together in a specific way. These subunits cooperate to bind and release oxygen efficiently. The forces holding subunits together are generally the same non-covalent interactions that stabilize tertiary structure: hydrophobic interactions, hydrogen bonds, ionic bonds.
So 4 levels describing this intricate architecture. And this structure is absolutely vital for function. And when it goes wrong?
When it goes wrong, it can lead to disease. A really dramatic example relates back to tertiary structure and folding prion diseases.
Prions, like mad cow disease.
Exactly. Mad cow disease, BSE, in cattle, Creutzfeldt-Jakob disease, CJD, in humans, scrapie in sheep. These are caused by infectious agents called prions.
What's so weird about them?
What's astonishing, and what Stanley Prusiner won the Nobel Prize for figuring out, is that prions appear to be purely protein-based infectious agents. No DNA, no RNA.
Just protein. How can that be infectious?
The prevailing theory is that a prion is a misfolded version of a normal cellular protein called PRPC. This misfolded form, called PRPSC, has the same primary sequence, the same amino acids, but its secondary and tertiary structure is drastically different, often with more beta sheet content.
Okay, so it's folded wrong. How does it spread?
This is the truly insidious part. The misfolded prion protein, PRPSC, can somehow induce normally folded PRPC proteins to misfold into the PRPSC form. It acts like a template for misfolding.
So one bad apple spoils the bunch.
Essentially, yes. This triggers a chain reaction, leading to the accumulation and aggregation of these misfolded proteins, particularly in the brain, causing severe neurological damage. It's a terrifying example of how critical correct protein folding is.
Wow, structure really is everything. Now, aside from causing disease when misfolded, what are the main functions of proteins?
Oh, the functions are incredibly diverse. We can broadly classify them by shape first. Fibrous proteins like keratin or collagen are typically long, linear, and provide structural support or rigidity.
Like keratin in hair and nails you mentioned.
Right, and nails have more disulfide cross-links in their keratin, making them harder than hair. Then you have globular proteins, which are generally compact, spherical-ish, and often soluble. These are the workhorses involved in metabolic functions.
Like enzymes.
Enzymes are a huge, critical class of globular proteins. They act as biological catalysts, speeding up virtually every chemical reaction in our cells, sometimes by incredible factors like 10 to the power of 17.
Trillions and quadrillions of times faster.
It's mind-boggling. They do this by binding specifically to the reactants, substrates, and stabilizing the transition state of the reaction, which lowers the activation energy needed for the reaction to occur. Our bodies rely on tens of thousands of different enzymes just to function.
Amazing. What other functions?
Transport is another big one. We talked about hemoglobin transporting oxygen. It binds oxygen via a non-protein helper group called hemogram, which contains an iron atom. Hemoglobin carries O2 from the lungs to the tissues.
Right, and this ties into sickle cell anemia.
Yes, tragically and powerfully. Sickle cell anemia is a genetic disease caused by a tiny change in the primary structure of hemoglobin.
How tiny?
Just one single amino acid substitution in the beta-globin chains. At the 6th position, a polar glutamic acid residue is replaced by a nonpolar valine residue. Just one change out of 146 amino acids in that chain.
And that one change causes the disease.
Yes. That single substitution creates a hydrophobic, sticky patch on the surface of the deoxyhemoglobin molecule when it's not carrying oxygen. In low oxygen conditions, these sticky patches cause the hemoglobin molecules to clump together, forming long, rigid fibers inside the red blood cells.
Which distorts the cells.
Exactly. It forces the red blood cells into that characteristic crescent or sickle shape. These sickled cells are fragile, break easily, and can block small blood vessels, leading to pain, organ damage, and the severe symptoms of the disease.
It's just staggering. One amino acid change.
It's perhaps the most dramatic illustration of the principle. Primary structure dictates tertiary and quaternary structure, which in turn dictates function. A tiny change, the most fundamental level, can have catastrophic consequences for the entire organism. It really underscores the precision involved.
So, wrapping this all up, what's the big takeaway here?
Well, we've gone from these relatively simple amino acid building blocks
Through how they link up into peptides.
to the complex hierarchical folding of proteins into specific 3D shapes.
And it's that precise structure at every level that dictates everything, whether it's carrying oxygen, catalyzing a reaction, providing strength, or even, when it goes wrong, causing devastating disease.
Exactly. From a fingerprint trace using 9-hydrogen to the intricate dance of hemoglobin or the misfolding nightmare of prions, it all comes down to the chemistry and structure of these incredible molecules.
It really makes you appreciate the molecular machinery inside us.
Absolutely. And maybe it leaves us with a thought. Given how sensitive function is to structure, how subtle changes can have such profound effects,
Yeah.
what else is going on at this molecular level that we're only just beginning to understand? How much more intricate is the architecture of life?
A fascinating question to ponder. Thank you for joining us on this deep dive into amino acids, peptides, and proteins. We hope you feel a little more well-informed, maybe a bit amazed, and definitely more curious about the incredible chemistry that makes life possible.